My name is Tyler Jacks. I'm the director of the Koch Institute for Integrative Cancer Research at MIT, and it's my pleasure to welcome you to the 10th annual Koch Institute Symposium. This is an important day for us each year where we bring together cancer scientists and others investigating the disease in new and emerging areas that we find to be particularly important, and highlight some of the important advances that are also taking place here at MIT. And these symposia have covered a range of topics from metastasis and nanotechnology, personalized cancer medicine, and systems biology. And today is no exception to the important themes with cancer metabolism being the topic of the day. And that's, of course, a very important area in cancer science. And I'm very much looking forward to the presentations from our speakers today, and I want to thank them all for their contributions in advance. It's also an opportunity for us to highlight some of our own scientists, and we'll be hearing both from David Sabatini and Matthew Vanderheiden today in the program. And we also use this as an opportunity to invite back former trainees each year, and this year that role is filled by Marcia Higas, who's a faculty member at Harvard Medical School and was once a postdoctoral fellow with Lenny Guarente here at MIT. So again, I'm very much looking forward to today's program, and I'm sure you are as well. Some of you will remember that last year we had a special event symposium. It was a 2-day instead of a 1-day, and it was a reunion event where we invited back all of the past trainees from the former MIT Center for Cancer Research and used that as an opportunity to allow folks to revisit their old home, which would only be the home of the Cancer Center and then Koch Institute for another several months, because we were intending to move on from there to our new home later that year. And of course that has happened. We have formed the Koch Institute for Integrative Cancer Research. The goal of the Koch Institute, as I hope all of you know, is to bring together cancer scientists and engineers to develop new understanding and new technology to solve the long-standing problems of cancer. And this is an explicit effort to bring together biologists and engineers under one roof. And the goal really is to integrate and collaborate across disciplines to both advance cancer discoveries, but also to advance new solutions to the long-standing problems of cancer. And as we had hoped would be the case, we did indeed finish the building. It was actually finished on time, slightly ahead of schedule, actually, and slightly under budget, which is frankly remarkable in this day and age. And we moved in late November, December, and then dedicated the building in a couple of day ceremony in March, which caught a lot of attention locally and actually nationally as well, including this front page story in the Boston Globe, which featured not only what we were doing in the Cancer Center Koch Institute, but also the contributions of the main philanthropist behind this effort, David Koch, and some of the special features of the new building as well. I want to point out quickly that this business here of the doctor indicted for 6 drug fatalities had nothing to do with us. Pretty sure it was a doctor across the river, but this was a fun event, a fun couple of days, and one particularly funny moment for me was I was walking along the big tent in the courtyard behind the building, and there was a tour group coming through, as is often the case here on the MIT campus, and the tour guide was explaining the festivities to her group of tourists, and she said, yeah, they're dedicating that building over there, which was given by Coke or Pepsi, I don't know what it was, but in fact, the individual who made it all possible was David Koch, shown here at the festivities, sitting next to the other person who made it possible, who is Susan Hockfield, our president, who has been a huge supporter of bringing together life scientists and engineers across the campus, not limited to cancer, but certainly as it results to the new Koch Institute project. And so in past years, I've shown you artist renderings of what the building would look like. Now I can actually show you what it does look like. This is an actual photograph, slightly photoshopped, I must say. For those of you who are around here, you will notice that the street light has been removed for photographic effect. But the building is lovely, and the move was very, very successful. We didn't have any major downtime. All the systems worked well, and the layout and the plans that we worked so hard to achieve were also clearly working well in terms of fostering our science and fostering integration. As I mentioned, the building has a mix of scientists and engineers, and these are the cancer scientists who work in the Koch Institute, the cancer biologists, I should say, from the Department of Biology, and they have been joined by an equal number of talented engineering faculty drawn from across the MIT campus, whose names and affiliations are shown here. And you can see that it's a diverse group, individuals who come from chemical engineering, material science, mechanical engineering, and other engineering disciplines. And we've settled in. Each of the floors of the building actually has a mix of cancer scientists and engineers working together. And many of the elements of the design of the building really do promote interaction and cause individuals to bump into each other and start that conversation that sparks the new idea. And we're actually now seeing the fruits of that playing out in real time, which is really very exciting and very satisfying for all of us. Those of you who are not part of the MIT community may not know exactly where the building is. It's on the eastern edge of campus, not far from here, and it's in a terrific neighborhood surrounded by a lot of other very important buildings at MIT and in biomedical research, including the Whitehead Institute and the Broad Institute, the biology home building, chemical engineering, biological engineering, and many more. So we really do feel like we're at the hub of a very important part of our campus. And important to our mission is to reach out and interact with colleagues, not just in these buildings, but really from across the campus. And in fact, we have extramural members of the Koch Institute who almost equal in number the individuals whose laboratories are in the building, and we hope to grow that network still further in the coming years. I won't spend a lot of time this morning describing the Koch Institute, but just to remind you, we have 5 areas of research that capture much, not all, but much of the activity within the research laboratories, including nanotechnology, nanomedicines for cancer, new devices for cancer monitoring and imaging and tracking, a major program in metastasis funded by the Ludwig Foundation, and a group of investigators interested in personalized cancer medicine and drug resistance mechanisms, and another group of investigators interested in cancer immunology and how to use the immune system more effectively to control cancer. And again, these projects are represented by individual research in the individual laboratories, but also in every case, collaborations between laboratories and between disciplines. And collaboration is really at our core. It includes interactions between cancer scientists and engineers in our building and around MIT, but also with other entities, including, importantly, the National Cancer Institute. We are an NCI-designated cancer center and receive a lot of support from the NCI. We also have important interactions with industry. In fact, just yesterday, we had our inaugural Science Exchange Day with Johnson & Johnson, which is a corporate partner now of the Koch Institute. And we feel that these interactions are a very important part of our mission to move our discoveries and our new technologies out from our laboratories where they can most rapidly benefit cancer patients. And along those lines, we do want to focus on patients, and because we don't have a hospital here and we don't take care of cancer patients here at MIT, it's important for us to reach out to other centers that do, and we're fortunate that in Boston we have many such centers, including the Mass General Hospital and the Dana-Farber Cancer Institute, where we have already established collaborative interactions with cancer scientists and clinicians from their laboratories working with scientists and engineers from our laboratories. And this is very exciting for us to see. And recently, just at the end of last year, we formalized this project under the heading of the Bridge Project, which is a formal interaction between the NCI-designated cancer center here at MIT and the NCI-designated cancer center at Harvard, which is called the Dana-Farber Harvard Cancer Center, and it actually includes Dana-Farber, MGH, Brigham and Women's, and in fact 7 entities that joined together under the heading of the Dana-Farber Harvard Cancer Center. And so with the Bridge Project, we hope to attract even more great ideas that will bring together scientists, engineers, and clinicians, clinical investigators, to bring discoveries and technologies as quickly as we can to new solutions. So before I close, I will just show you a few more images. I've shown you this one from the outside, and I encourage you, if you haven't been to the Koch Institute already, to come by for a visit or, in fact, a tour. The laboratories are very nice, and also on the first floor, we have a lot of elements to sort of introduce you to the development of the Koch Institute with respect to a long timeline of advances in science and engineering here at MIT and noteworthy discoveries shown in this part of the entryway. Just under that on the floor is a mosaic which shows the position of the Koch Institute with respect to the MIT campus. And then along the corridor, which runs along Main Street, is a series of art and educational exhibits, all based in science, to teach you about the kind of work that we do and hopefully inspire the next generation of cancer researchers. This is actually a mural created by some Media Lab trained artists based on data generated from Mike Yaffe and Forrest White's laboratory. It's really quite interesting and I think quite beautiful. These are some science display cases that you might find otherwise at a science museum to teach specific stories about research that's taking place at the Koch Institute. And further down the corridor is a series of large format prints of images from our laboratories and others actually that highlight really the very interesting and very informative things you can do with various types of imaging technology. And again, the goal here is to capture people's attention, capture people's imagination, and hopefully inspire them to get involved in this kind of research. And importantly, this part of the building is open from 6 a.m. to 6 p.m. Anybody can walk in and experience and hopefully enjoy these exhibits. And so I'll stop there and encourage you to stay abreast of the developments in the Koch Institute, stay involved. You can log on and read about new things happening in the Koch Institute from our website. Again, I thank you for being here this morning and participating in this symposium. And now I'll turn things over to Matt Vanderheiden. Matt was one of the two organizers this year, and I appreciate very much his contributions to putting the program together and organizing today's events. He was joined by David Sabatini in that effort, and I'm appreciative to him as well. And I also want to thank a very large number of people actually who contributed often behind the scenes to make this event possible and to arrange all the various things that will happen today. Cindy Quentz and the whole headquarters office, the executive director's office, all the administrative staff have pitched in wonderfully, as they do every year, to make this symposium successful. And this year I'd like to especially thank Laurie Spindler and Sarah Hufford, who went out of their way and really went above and beyond their normal responsibilities in the Koch and devoted a tremendous amount of effort to make this event so great. So let's take a moment to appreciate them for all that they've done. And with that, I'll turn things over to Matt Vanderheiden, Assistant Professor in Biology in the Koch Institute, to take it from here. Matt.
Excellent. Thank you, Tyler. So thank all of you for coming. If my talk is up here, I have my own set of thanks that I'd like to give. If you guys can hear me back there, can you put my slides up, please? Okay, thanks. There's also a number of people who I would like to thank. First off, Tyler for helping to bring this symposium together and choose the topic that's very near and dear to my heart. We have several people to thank who were instrumental in putting the funding together for this event. One is that we've had a number of vendors who have contributed to making this symposium possible, and I would encourage all of you to please visit their tables out in the lobby during the breaks today, and in addition to that, to do your best to support their various products. There's a number of corporate sponsors who have been very important in the educational mission of the Koch. In particular, because of their generosity, we were able to pull together such a wonderful group of speakers today and put on such a nice event. I would also like to thank a number of people who were particularly helpful to me in putting together the scientific program. First off, David Sabatini for numerous discussions about actually putting together what I think is a fantastic list of speakers, and thank each of you for joining us here today. And Laurie in particular for really stepping up and taking on sort of the duties of most of the behind the scenes work, with particular help from Sarah, Pam, and Cindy, who all were great in terms of pulling things together. So thank all of you. Now, I'd like to just get started to sort of kick today off by giving you guys a general background with at least my perspective about why we're here today. Metabolism is one of these fields that has been around for a very long time, and why are we thinking about it now in terms of cancer? And most of you are probably aware that, in fact, metabolism was one of the first differences that was ever noted between cancer cells and normal cells. This predates the discovery of DNA and knowing anything about the genetics of cancer. And it was really back in the 1920s that the work of Otto Warburg first sort of pointed to metabolism as something going on in cancer. And he was really following up on the work of Louis Pasteur. So Louis Pasteur, as most of you know, discovered fermentation. And what Warburg was doing was essentially repeating Pasteur's experiments in normal tissues as well as in tumor tissues from the animal models of cancer that existed at the time. And what he found is that normal tissues tended to generate lactate only when oxygen was absent, and when oxygen was present, there really was not much lactate production at all. This is really the mammalian correlate of Pasteur's fermentation experiments. And what he found is that tumor tissues tended to make lactate regardless of how much oxygen was there. And this is now what is often quoted as this Warburg effect that I'm sure you'll hear about from a number of the speakers today, and certainly we'll read about in various journals and articles on cancer metabolism. Now, in modern terms, we know what Warburg was really looking at was the difference between how glucose is metabolized. And as we all learned in undergraduate biochemistry, you know glucose can be metabolized by glycolysis to pyruvate, and that pyruvate can be completely oxidized in the mitochondria in a process that requires oxygen, molecular oxygen, as the final electron acceptor. Now, because oxygen is required here, to get this high number of ATPs, 36 is the number that I was forced to memorize as an undergraduate, you need oxygen, and so when oxygen is not there, you can't do this process. However, you can still metabolize that glucose to pyruvate, but then that pyruvate has to be converted into something else. In the case of mammalian cells, that's lactate, and that's necessary in order to generate the NAD cofactor to allow glycolysis to continue. And as we, again, learned, this is a less efficient process, at least in terms of ATP production. Now, Warburg, of course, noted that cancer cells tended to do this anaerobic process regardless of whether oxygen was there, and hence this Warburg effect is also referred to as aerobic glycolysis. Now, we've known about this now for almost 90 years. It's going on 90 years here in the next decade. And as a result, it's actually quite surprising that even today, we still don't fully understand what regulates this, nor do we understand truly what the importance of this change is for cancer cells. Now, I think it's also fair to point out that many people view this as an all or none phenomena. It's very clear that all cells, to a certain extent, will make lactate. In addition, that cancer cells actually require mitochondria, and I think we may hear some ideas about this during some of the talks today. I think you'll also learn during the talks today that there's many other metabolic changes that go on that really extend beyond this Warburg effect, and I think you'll find today during the talks a lot of interesting information about what is really the state-of-the-art knowledge about how these changes in metabolism might be influencing cancer growth. Now, for a number of reasons, cancer metabolism has sort of been, rediscovered by a number of people, and I think there's a few reasons for that. One is because of the clinical side, the popularity of FDG PET scanning has really grown in recent years. Now, this is a technique where glucose is labeled with a radioactive isotope, and that glucose analog is taken up by cells, phosphorylated and trapped there, and this can be imaged with using PET. And it turns out that this is a great way to label many types of cancers. And so as a result, FDG PET scanning is quite popular, mainly for cancer staging, but sometimes to work out things regarding diagnosis. Now, what's become increasingly clear over the last several years is that not only is PET a good tool for diagnosis and staging, but it can also be predictive. And that is that regardless of various therapies, studies have shown that in many different tumor types, the ability to make a tumor go negative by FDG PET scanning seems to be as predictive as anything else for ultimate response to therapy. And this is two examples shown here. This is our gastrointestinal stromal tumor that's treated with a tyrosine kinase inhibitor, imatinib. This is a patient with lymphoma who's treated with standard chemotherapy, CHOP Rituxan. And both of these patients received this therapy, had a PET scan shortly after being given the therapy, and as you can see, the tumor in this case has gone PET negative. In this case, the tumor has gone PET negative, and this was really a finding that existed long before any other sort of standard imaging techniques could detect a response to these tumors. And so increasingly, because of this, clinicians have become more and more interested in understanding what's going on with this technique, if indeed it is predictive for a response to therapy. In addition, basic cancer scientists have for a number of years been studying a number of these various genetic mutations that drive cancer, things such as KRAS, PI3 kinase, MYC, and among the many things that these various signaling proteins do to drive cell proliferation, it's become increasingly clear that they all talk in one way or another to central carbon metabolism. And so that's raised a lot of interest to really understand what is this convergence of cancer signaling pathways on metabolism telling us about the biology of this disease. Now, many of you might say, well, don't we know a lot about metabolism? This is, the chart that I have on the wall in my office. And, we do know a lot. But as someone who teaches undergraduates biochemistry, I can tell you that most of what we teach in biochemistry classes is really about how we produce ATP. And I think you'll learn today that there's still a lot to be discovered about how metabolism is regulated, how nutrients are sensed, new techniques coming on to image metabolism, both at the cellular level or at the whole organism level. And we're going to hear a number of talks today. Now, in the classic Weinberg and Hanahan hallmarks of cancer, you'll be excited, at least the speakers will be excited, that we've been elevated to a full emerging hallmark at this point. And so I think you'll hear from a number of the speakers today why that is the case. And so I'm very excited about today's program, and I thank all of you and the speakers for coming, and I look forward to an exciting day of science. A couple logistical notes to make. There are 2 breaks during the day. Per Koch Institute tradition, we will not have individual question and answer sessions after the talks today, but I encourage the speakers to make themselves available during the breaks to address any questions that might arise, and of course also use those break periods to visit our various vendors' tables out in the lobby. And with that, I'm going to turn things over to Jackie Lees to introduce the first session.
My name's Jackie Lees. I'm one of the associate directors in the Koch Institute, and I would like to join Matt and Tyler in welcoming you to the symposium today. So this session is on metabolic regulation, and the first speaker is David Sabatini, my colleague. So David gained his MD PhD at Johns Hopkins in 1997, and it was actually here that he began to study the role of mTOR, which is really his life's work. He then became a Whitehead Fellow and continued that work, and in 2002 became an official faculty member at MIT and a member of the Whitehead Institute. He is also an investigator of the Howard Hughes Medical Institute. And his lab is generally interested in cell growth and the metabolism of cancer, and as I said, he's particularly well known for his contributions to the field of the mTOR pathway. And the title of his talk is mTOR Signaling and Cancer Metabolism. David.
Okay, thank you, Jackie, for the introduction. Also, I want to personally thank Matt Vanderheiden, really, for being the brainchild for most of the invitations and actually for doing the majority of the work. It's always a pleasure to do something with an assistant professor who goes on and does so much work. I also want to thank personally all the guests, the speakers who came, and all of the attendees. So we've really been interested in three areas. As Jackie mentioned, one is the regulation of growth, mostly through the TOR pathway. We've also done a lot of technology development, largely in RNAi libraries. And much more recently, we've also become interested in cancer metabolism, largely because of the realization that this pathway, the TOR pathway, is a major sensor of metabolites and nutrients and also a major regulator of metabolism. So I'll tell you two stories, one on amino acid sensing by the TOR pathway and another one on amino acid metabolism in the cancer metabolism field. So really, amino acids or connect both of these. So despite really the obviousness of size variation as a feature of biological systems, as shown here, we actually know relatively little about what controls the sizes of cells, much less about what controls the size of organs and body size. The TOR pathway, along with the Hippo pathway, are turning out to be the main regulators of growth in all eukaryotes. Now, insight into the TOR pathway comes, as many of you will know, from kind of an interesting angle, and that is from this small molecule rapamycin. I would argue this is one of the more interesting molecules out there. It's a natural product. It's made by bacteria, found originally on Easter Island, where you find these very interesting statues that people don't really understand their significance. And this molecule has had profound clinical impact, as shown here in immunology, in cardiology, and increasingly in cancer. It's also led to the discovery of the TOR pathway, which is turning out to be really a fundamental, basic pathway that operates in many, many different physiological processes. Now, rapamycin was sort of an obscure molecule, not in the science world, but in the sort of the popular press. And in the last few years, there's been sort of an explosion of interest in it, with the finding that rapamycin prolongs lifespan really in many different model organisms, particularly even in middle-aged mice. And this is one of the few FDA-approved molecules that will do this, and quite a significant effect on lifespan. So since 2002, we knew quite a bit about this pathway. We already thought it was very complicated, and this is a slide that I pulled out from back then. At this point, we already knew that mTOR was the central component of this pathway. This was perturbed by rapamycin, as I'll mention later on. We also knew that mTOR was a regulator of the balance between anabolism and catabolism, so an inhibitor of catabolic processes such as autophagy, as we'll hear later on, and also a regulator of anabolic processes, for example, protein synthesis, ribosome biogenesis, and increasingly there's interest in the transcriptional regulation, which we now largely know is metabolism-driven, either through the HIF pathway or the SRBB pathway. The feature of this pathway that's always fascinated us, and I still think is the most interesting aspect of it, is that this pathway, unlike most signaling systems, pretty much senses everything. So all kinds of nutrients, energy levels, different stresses, all types of growth factors, oxygen levels. And so this tells us the cell cares a lot about connecting the environment to the activity of the TOR pathway. And we've been particularly fixated on understanding how this actually happens at a molecular event and how these events can actually be integrated to give coherent growth rates and therefore cell size increases. Now, since this time, there's been an explosion of knowledge in this pathway. We know that this original pathway that we called mTOR pathway is now actually the mTOR complex 1 pathway, or mTORC1. I'll talk a little bit more about this. And the realization has been that mTOR doesn't act alone. It actually is part of large multi-protein complexes within the cell, which define mTORC1 and a related complex that has some similar proteins in it, but some different ones as well, that we now call mTORC2, or mTOR complex 2. I'm not going to talk more about this pathway, but this turns out to be part of the PI3 kinase pathway, and one of the kinases that activates AKT. Now, both of these complexes are intimately connected to cancer. In the case of mTORC1, there are many mutations that were first discovered in cancer-prone syndromes, so relatively rare diseases, but increasingly are being found in common cancers through cancer genome sequencing efforts. And so really you can see mTORC1 is surrounded by these tumor suppressors and oncogenes. And mTORC2 is part of the PI3 kinase pathway, therefore is part of one of the most commonly mutated pathways in cancer. This has led a drive there to develop inhibitors that target these complexes. The original inhibitors are really rapamycin, which we call the limus compound. Sirolimus is another name for rapamycin. And there's a number of these. These are all, in essence, derivatives of rapamycin. And these are molecules that now, in hindsight, we realize are quite complicated. These are allosteric inhibitors of mTOR, and they partially inhibit mTORC1. They don't fully inhibit its activity. And in fact, in certain cases, can even inhibit mTORC2. This has led a drive to develop a second set of inhibitors. These are much simpler to understand. These are all ATP-competitive inhibitors that target the kinase domain that's shared by both of these complexes. And you can see there's a number of these different inhibitors, some which are more or less specific. Some also target PI3 kinase. These are likely to be more effective, this class of inhibitor, but also are likely to have much more toxic effects, which rapamycin, surprisingly, is a relatively benign molecule. So let me show you a little bit about the growth control by mTORC1 in vivo.
what you're looking at here is a mouse liver. You can see here's like a normal liver. Here's next to it is a liver in which we've inhibited mTORC1 genetically. You can see it's about 40% smaller, and pathway activity is down. And conversely, here we've activated the pathway, and the liver is larger, and pathway activity is up. Many of you know that if you fast an animal overnight, a mouse, it'll lose about 25% of its liver mass. A 2-day fast, you'll lose about 40% of its mass. And you can see here, when we inhibited mTORC1, there's no further decrease in the size of this liver, indicating that in response to these nutritional cues, this liver is as small as it's going to get. Conversely, when we constitutively activate this pathway, there's really a resistance to these anti-size effects of fasting. And in this case, this is really an active degrowth, probably an autophagy-driven degrowth of the liver. So if you think about these hepatocytes in vivo, there's really two types of messages, or two types of signals that they have to respond to make these types of decisions as to whether to grow or not, or to actively degrow. One of them, of course, is nutrient levels, because no cell should be stupid enough to try to grow or in the absence of the building blocks for both mass as well as energy. And of course, this is why unicellular organisms use nutrients as their main drivers of growth and proliferation. The other signal is more particular to multicellular organisms. They need to have an idea as what the entire body is doing. Therefore, there are hormonal signals that have evolved to transmit to all the cells throughout the organism that the organism itself is in the right state for growth. And insulin is the best known of these, but there are a number of these. So as a growth pathway, then, for you, one needs to integrate both of these classes of signals. And so we've been interested in how does the TOR pathway do this, and we know quite a bit about this. A lot of this integration happens, for example, from energy levels, oxygen, growth factors, mitogen. It happens at the level of a tumor suppressor called TSC. And through unknown ways, it integrates these mechanisms. TSC is a negative regulator of a small G protein called Reb, and Reb will bind when it's GTP loaded to mTORC1 and strongly activated. And in the absence of Reb, there is really no activity of this pathway. So it's a key component of this system. The signal that's not shown here, and we've been particularly interested in, is amino acids. This is probably one of the primeval signals that this pathway evolved to sense. In the absence of amino acids, none of these things work, but amino acids do not go through TSC. And so the question is, how do amino acids work? So you could first ask, well, how about known amino acid sensing pathways? Do they impact this system? And of course, there are some. For example, neurotransmission relies on glutamate and glycine receptors and other amino acid receptors. However, evidence suggests that this pathway does not sense extracellular amino acids, so we can pretty much ignore these. There are intracellular amino acid sensing pathways, such as the GCN2 pathway that senses uncharged tRNAs, and here again, genetic studies show that this is not relevant. The important step for us came from the identification of these proteins that we found biochemically bound to mTORC1. These are small GTPases. They're part of the RAS family, although very poorly related to RAS. They're much bigger than RAS. They're about twice the size and have a domain that we don't know what it does. And they actually come in 4 different varieties, A, B, C, and D, in mammals. And what's interesting is that A and B are very similar to each other, almost identical, and C and D are very similar to each other. And for these to be functional, they have to form a heterodimer where there's an A or a B or a C or a D. And we use A and B interchangeably as well as C and D. And these proteins turn out to underlie what we think is an amino acid sensing pathway that is having quite a few different sort of twists and turns that we think are interesting. So when there's no amino acids, the B form has GDP bound, and the D form, or C, has GTP. You add amino acids, and this flips through a mechanism that we don't understand how it works. And importantly, it binds now strongly to mTORC1. And I'll show you a little bit of the data here. You can see minus plus amino acids. There's an interaction when there previously wasn't one. And if we put in cells a mutant of B that can't hydrolyze GTP, so it's a constitutively active B by being always bound to GTP, you can see that there's no longer regulation. Moreover, this mutant, if you put it into cells, will make the pathway insensitive to amino acids. That is, that it will always be constitutively on, even if we remove amino acids. So we're a little concerned that we're overexpressing things there, and so we generate knock-in MEFs, or knock-in animals, actually, where we've put into the RAG-A locus. Both alleles are this GTP form. You can see in control MEFs, there's nice regulation of this pathway if we look at one of the canonical outputs. But in cells where we've knocked in it's a RAG-A GTP MEFs, there's no regulation. These animals, unfortunately, I don't have time to show you more data on them. These animals die perinatally. They actually don't have developmental defects, and they share quite a few similarities to animals that are defective for autophagy. So one of our leading hypotheses is that these animals have an issue when they're born because they, in essence, starve to death before they suckle from the mother because they can't induce autophagy, although that remains to be proven. So in a variety of different experiments, we've gone on to show that these proteins are necessary and sufficient to mediate amino acid signaling to mTORC1. But the difficult part has been understanding how they actually work, because the obvious idea that these are simply binders of mTORC1 that activate kinase activity turns out not to be the case. The big insight came from when we looked at the localization of mTOR. I'm not sure you can see this too well here, but in the absence of amino acids, mTOR is in this sort of fine speckled pattern that we actually don't know what this represents. When you add amino acids, though, there's this really dramatic, very fast translocation to these vesicular structures that are perinuclear. And I'll show you that this is truly a translocation in this movie. When we add amino acids to this white box, you can see the appearance of these puncta that are first small and then become larger and coalesce. And so we said, well, the RAGs don't affect mTORC1 kinase activity, that is specific activity. Do they affect its localization? The wild-type proteins, if you overexpress them, they do not. However, if you put in this B form that's constitutively loaded with GTP, and you may recall I said this protein makes the pathway insensitive to amino acid starvation, you can see the appearance of these structures even when there's no amino acids present in the cell. And so this was a correlation then between this localization and the activity, and ultimately we went on to show that when we add amino acids, mTORC1 moves to the surface of the lysosome. That's what these structures turn out to be, so not inside, but on the outside of the lysosome, where the RAG proteins, I didn't show you this, are always there. The RAG proteins are actually quite good markers of lysosomes, again, on the surface of the lysosomes. So how does this connect to the rest of this pathway? Well, the important insight was that Reb is also part of the endomembrane system and also on endosomes and lysosomes. And so we proposed a model where when there's no amino acids, Reb and the RAGs on the lysosomal surface, mTORC1 is in some other unknown compartment. We add amino acids, mTORC1 translocates the RAGs using them as a docking site and can then encounter this activator Reb. And we very much liked this model because this is in essence an AND gate. You need amino acids to put mTORC1 next to its activator, but you need all these other inputs for Reb to be loaded with GTP. And this could explain why, when you remove amino acids, none of these signals actually work anymore. Now, to prove this model, and I won't share the data for this, what we've gone on to do is to basically force mTORC1 onto the lysosomal surface by putting lysosome targeting sequences that put it on the surface. And if you do that, the pathway becomes insensitive to amino acids and actually no longer cares about the RAG proteins. They can be deleted. So this model has led to a number of different questions. One of them that we're really struggling with, or I am struggling with, is why these are heterodimers. There's really no reason from what I've told you so far, yet they are. Is this system deregulating cancer? All of these have oncogenes or tumor suppressors, that is, these pathways, so we might imagine there would be one here, but we have yet to find that. And then what is likely, I think, the most interesting question, what is the amino acid sensor and where is this sensor? So we don't know what it is, but we're starting to think where it is. We know where it is. And that's because in the last year, we identified another complex that goes by this name, Ragulator. Many people snicker when they hear that name, but that's what we ended up calling it. This is a heterotrimeric complex. It's also essential for the activity of this pathway and is also on the lysosomal surface. As soon as we purified this, we knew it had to be part of the mTOR pathway because there actually is a mutation found in people that partially decreases the level of expression of one of the proteins of the Ragulator. And this is a family that has this mutation. These are 4 children from a large consanguineous family. And they came to clinical attention because they had immunosuppression, which sort of piqued our interest because rapamycin is an immunosuppressant. But what really got our interest is that if you look at the growth curves for these children, they're all incredibly small. So they're all at the 3 to 5% in size. And of course, the mTOR pathway is a growth regulator pathway. In addition, you'll notice that they're actually quite young looking. So this boy here is 18, and he certainly does not look 18 in this picture. And so again, these sort of anti-aging effects were interesting. And so we went on to show that the Ragulator is a key part of this pathway. It actually is a scaffold that holds the RAGs on the lysosomal surface. And so the picture then that's emerging is that amino acids have a special role, that lysosomes have a special role in this pathway in amino acid sensing. And this was quite controversial when we first proposed it. But I think in retrospect, it makes quite a bit of sense because autophagosomes, of course, fuse with lysosomes and to release their contents there, and amino acids are processed there. mTOR is a regulator of autophagy, so it might make sense to have it at that location to sense the products of autophagy. In yeast, amino acids are stored in the lysosome. And so we're operating on the idea that in response to amino acid levels, some feature of lysosomes changes that then is transmitted as an amino acid sensing mechanism to the mTOR pathway. Now, one could ask, well, where exactly are amino acids sensed? We know they're not sensed on the outside, but it's actually very hard to decipher, are they sensed on the outside of the lysosome, are they sensed in the inside of the lysosome when you're working with intact cells. So there are really 2 obvious places, on the outside or inside, and all of these proteins, despite the way they're shown here, are attached to the outside of the lysosome. They're not transmembrane proteins. So to get at this, we've recently developed an in vitro system where we can study this. We can take purified lysosomes, purified mTORC1, mix them in vitro, add amino acids, and observe this translocation. And one important thing this tells us is that everything for sensing is contained within lysosomes, because we can get very highly pure lysosomes, of course not 100% pure. So we've gone on now to test a couple of hypotheses using the system. I'll give you just a little summary of this. So you could ask, well, does the membrane actually even matter, right? So do you need this? And it turns out you do. If you dissolve it or you make holes in it in a variety of ways, that you have no more amino acid sensing. Do you need amino acids in the inside? You do, and there's a number of ways that we have tested this, so they have to get in to be sensed. Do you need the vacuolar ATPase, which makes the proton gradient that characterizes lysosomes? And the answer there is yes. The odd part, though, is you actually don't need the proton gradient itself. So you need the VATPase, but not the product, or at least the known product of the VATPase. And so we're operating on the idea that there's an inside-out amino acid sensing mechanism that requires this complex in a way that does not require its sort of known output. And so we're obviously chasing this down and trying to use this system to figure out exactly what the sensor might be and what exactly might be sensed. Okay, I'm going to shift gears here a little bit to what you might associate as being more traditional cancer metabolism work. And I've started here with, again, a slide that's reminiscent of what Matt showed you on the Warburg effect. And I won't go into all the Warburg effect at all. And of course, this is likely the origins of this field. But I'm just showing you this to say that when we decided to work on cancer metabolism, what we knew is that we did not want to work on the Warburg effect. That was our #1 conclusion, because there's really too many people working on this already and doing too much good work. Luckily, as Matt showed you, there's a lot of other pathways that you can see here. We spent quite a bit of time sort of identifying genes onto this pathway. We have come up with about 2,800 metabolic enzymes as well as transporter genes. I think I showed this in a talk that Craig Thompson was at, and he got upset. He said there's actually 4,500 of them, so maybe he'll show us later on. But any way you slice it, this is a remarkably large fraction of the genome, if there's around 22,000 genes. And we've been pursuing very basic questions around these pathways. So we want to know which of the metabolic genes and pathways are needed for cancer cells to proliferate and survive. That is just the parts list. And we want to know this in vivo. We want to know it in vitro. We want to know it versus different genotypes. And the approaches we're taking are also quite clean slate approaches, genomic approaches, looking at amplifications, deletions, gene expression patterns, and most importantly, loss of function screens, usually RNAi screens, but also increasingly haploid cell screens. So I'm going to tell you a little pilot screen that we've done that was intended to sort of test out the approaches, but at the end actually gave an interesting story. And so Rich, who started this work, did a bioinformatic analysis in breast cancer for genes that fit a number of different categories, for example, found in breast cancer or in any cancer versus normal or associative stemness. And you can quibble about whether these categories make sense, but this is actually what was done. And he basically crossed these gene sets and came up with a set of 133 genes that we thought could be enriched for genes that were important in breast cancer tumorigenesis. And so we generate RNAi libraries against all of these genes. These are lentiviral shRNA libraries. And really the most important thing that Rich did is he identified a breast cancer cell line shown here that has a very high tumor initiating capacity. Because of course all of you will realize that to do an in vivo screen and to be able to screen multiple shRNAs, many of the cancer cells need to contribute to making the tumor. If you put in 1 million cells and only 100 actually contribute to the tumor, you can only screen 100 things. And so this has a remarkably high tumor initiating capacity. So we made tumors with cells infected with these lentiviral shRNAs. We isolated genomic DNA, from the cells directly or after they were passaged in vivo after making tumors in the mammary fat pad. And there we basically compared by deep sequencing the frequency of shRNAs in the original starting set and in the cells that had gone through in vivo tumorigenesis, looking for shRNAs that were depleted. Presumably these are targeting genes that are important for tumorigenesis. Using a variety of strict criteria, we came up with this hit list, which is shown here. We were quite happy about this hit list because it has a number of genes that have already been implicated by others in cancer, so we thought that there was some enrichment for this. We ended up focusing on a gene called PHGDH. This is phosphoglycerate dehydrogenase, and I think you'll also hear from Matt a little bit more about this gene. Before telling you why we focused on this one, let me tell you what this is. This is encodes the first committed step in serine biosynthesis. It takes a glycolytic intermediate, 3-phosphoglycerate, and in a 3-step two-step pathway that involves a transaminase and a phosphatase makes serine. So serine is technically a non-essential amino acid because we can make it. Serine is also a special amino acid because in addition to being involved in protein synthesis, it also contributes to many other important processes. So through glycine to one-carbon pools and therefore to nucleic acid synthesis. And then it's also a component of lipids that are found in the cell membrane. So the reason we focus on PHGDH is if you take all the hits that came out of our screen and you interrogate genomic databases for genes that are amplified, so you could look for deleted ones, but it wouldn't make so much sense versus this screen. If you look for genes that are amplified, there's only really one that fits the bill, and that is PHGDH. So it's on a peak in melanoma, quite a significant peak in melanoma, and on a less significant peak in breast cancer, which is where we had actually done our screen. Moreover, if you look at the expression of PHGDH in breast cancer gene expression data sets, it always correlates with features that signify poor outcomes. So for example, basal tumors, high grade, estrogen receptor, ER-negative tumors, which are much more poorly treated than ER-positive tumors, and then whether the patient has died of their disease or not. We developed an immunohistochemical assay against PHGDH. You can see its expression in breast cancers is really dramatically different comparing across breast cancers. And after quantitating a large number of these, one can see that really the highest expression is found in this class of breast cancers, the ER-negative ones, that in general are poorly treated. In fact, we don't find a correlation with HER2 status in these studies. So for all these different reasons, we decided to study this gene a little bit more. One can actually verify that it's highly overexpressed in cells that have amplifications. This is a tissue culture experiment with a number of different breast cancer cell lines. You can see that there's higher levels of expression actually than you even expect from the amplification. So first thing you can ask is, well, is the activity of this pathway actually up in the cells that overexpress this? And in doing metabolic studies, one can actually find that serine synthesis flux is dramatically higher in these cells versus these. Of course, these cells have many genetic differences versus the ones that are non-amplified. And you can show that simply adding the cDNA for PHGDH to these cells converts them to having the same levels of serine synthesis flux than the ones that have the amplification. So PHGDH is really rate limiting for serine synthesis in cancer cells, which actually is different than what's thought in normal tumors, where the last step of the pathway is thought to be rate limiting. Okay, so you can then ask, well, do the cells that have the amplifications actually care about having this? And by using RNAi, you knock down this gene, you can see there's a dramatic effect on cell numbers in cells that are amplified. This is actually quite a dramatic effect on sort of morphology of the cells. You can see the cells look quite healthy with the knockdown. However, it does not appear to be apoptosis. We've tried to measure that, so we don't know the type of death that happens here. In cells that are non-amplified, actually, they're quite not affected by the loss of PHGDH, the low expression that they have. Now, many of you will already be thinking that this is a little bit strange because these are cells that are growing in tissue culture media, which of course has many, many nutrients, including many amino acids, and most common media has serine in it. And so here I'm telling you that a pathway that makes serine is essential in cells that are growing in the presence of serine, which doesn't intrinsically make so much sense. This is actually the case, though. Here, this is a similar experiment done in media that has 1x concentration of serine. We said, well, maybe there's not enough serine. Maybe it's sort of a partially essential amino acid. It's not the case. If you give them more serine, nothing happens. We measured serine transport. It's fine. We used cell-permeable versions of serine that can get in through transport-independent mechanisms, and we've shown can fully substitute for regular serine. Yet we cannot rescue this phenotype with serine. So that argues that serine is not the only important thing that this pathway is doing. And sometimes we lose sight of the fact that a pathway like this, that has multiple steps in which we know simply overexpressing this component leads to more serine, we then can know that for every molecule of serine, we must make these other intermediates, these other byproducts of the pathway. We looked at NADH levels, we really saw very few differences there. We ignored the inorganic phosphate, but alpha-ketoglutarate was actually quite tempting. This is, of course, a central metabolite in many different metabolic pathways, and for a number of reasons, we focused on it. So here's the pathway again. Here are the outputs of serine. The thing to keep in mind, and many of you who are aficionados of metabolism will know, alpha-ketoglutarate is a component of the TCA cycle, the Krebs cycle, and there is a phenomenon that undergoes in cancer cells that has been referred to as anaplerosis, where cancer cells, because they take intermediates from the TCA cycle in biosynthetic reactions, much more than normal cells do, they need to basically replenish these intermediates to keep the cycle going. There's quite a bit of this. It's that other transaminases, and this enzymatic step transaminase are known to contribute to anaplerosis. And so we examined whether the alpha-ketoglutarate produced here would be significant for going into the TCA. And data I will show you suggested about 50% of alpha-ketoglutarate actually in the cells that have amplification is coming from the pathway. About 10% of the carbon flux going to glycolysis is being deviated down this pathway in cells with the amplification, while it's only very few percent of cells without. That these cancer cells then have evolved this mechanism to do anaplerosis, while other cancer cells do different pathways. Okay, a couple of conclusions in the last few seconds about amino acid sensing by the mTOR pathway. For us, it's been a really important conceptual step to understand what's the signaling event in amino acid sensing, which is this translocation. We also are excited about this sort of inside-out amino acid sensing mechanism, although we don't know what the sensor is. And then the second part, I told you about PHGDH, which turns out to be amplified cancers and may serve as a drug target in those types of breast cancers. And lastly, I think this pathway serves an unappreciated role in contributing to alpha-ketoglutarate anaplerosis through the cycle. So in the first part of the talk, was initiated by who is a student who's gone on to better things now. She discovered the RAG protein, and together with Leon, they discovered the Ragulator. Roberto has done a lot of the work around the amino acid lysosome. Alejo generated the mice that I told you very little about. And then lastly, the metabolism part has been spearheaded by a postdoc who's worked with a large number of people in the lab on this. We've had key collaborators at Agios Pharmaceuticals, at the Broad, both RNAi screening and genomics, also by MGH pathology. So thank you very much for your attention.